Thank you very much. So it's a real pleasure to be presenting here. And I want to admit two things. The first one is what I'm presenting today. This is the first time I'm talking about it. So I really appreciate feedback. I know that in this weird Zoom world, I can't see everyone, but if you have any thoughts, I'd really be grateful to hear them. And the second thing is thank you, Arjun, because without paying him, he asked all the right questions, which lead right into my talk. So this is wonderful. So I'm going to take a step back, and I think that you've probably heard this many times before this symposium, which is the question of how do we order events in differentiation, and single-cell genomics clearly gives us a set of tools for thinking about this. And the starting point for us is really to take a high-dimensional data set. You can think about it as a matrix. We embed that data set. We can think about this as a high-dimensional sort of a point cloud in a high-dimensional space. And then we embed that data set in some low-dimensional space and hope that we can infer from here some continuum of curves from which we can make some statements about dynamics. And there are many strategies for just looking at those types of low-dimensional manifolds and learning from them. There's also ways of embedding information on local dynamics onto these manifolds. Here are just a few examples. For example, RNA velocity, which Philip just mentioned, and then several other methods which are going to give us the local direction or predict a local direction of change on one of these manifolds. You can also imagine just sequentially collecting cells over time, and that, of course, enforces a very sort of simple progression of time. And then the challenge is, of course, stitching together time points. And again, there's a collection of methods for that. So one of the challenges that we have is that even with this type of local instantaneous information, we still don't have a full view of what's going on in the dynamics. So there's still many ambiguities left. The simplest ambiguities, which many will appreciate, is simply the fact that there's many choices about how to do this embedding. So the pictures that we get are not unique in any way. Beyond that, even once we have an embedding and we think that it's very reliable, there's a question about how we should translate a local instantaneous dynamics into a long-term dynamics. And this is very easy to appreciate if one just considers a bifurcation where we don't know whether cells are initially uncommitted, and then when they approach a branch point, we'll decide to commit one way or the other, or alternatively, whether they were already strongly committed early on. And there's really nothing, even in the instantaneous measurements, such as RNA velocity, which can tell us simply which of these two cases is occurring, because we only have the average behaviors. So lineage tracing, and I think that Philip gave us a really nice example of this, it provides a way of establishing bona fide long-term dynamic relationships. And here's just another example, and I won't be talking about this very much. This is work that I've presented in the past and has been published, where we take an expressed random library of DNA barcodes, which are expressed in the 3' UTR of GFP under a strong promoter. We introduce this into cells with a lentivirus. This is very, very similar to work that Sam has really pioneered, her cell tagging work. Our method has the details are very, very slightly different. And we introduce these into hematopoietic progenitors, let the hematopoietic progenitors differentiate. And from there, we can, and again, this is looking at that same example that we looked at before, just focusing on monocytes versus neutrophils. We can look at cells early and then ask about what their sister cells are going to do later by resampling the same clone over time. And you can get a bona fide readout here of how the state of an initial cell predicts its fate. And if we now compare this to a set of computational methods which attempt to do this without access to lineage information, one sees that they perform in variable degrees. None of them perform perfectly well, and some of them perform poorly. And there's really no way a priori to know which one of them is right, until you actually look at the data. So this is sort of the key argument for why lineage tracing could be useful. Right, so that was just the simple way of reminding myself to say that these aren't great methods. And Philip also introduced his Linnaeus method, just to say that there's now really a whole family of methods out there which allow lineage tracing to be integrated with single-cell genomics. Some of them make use of Cas9 scarring, other ones make use of Cray recombination, other ones make use of integration of barcodes, and all of these in some way or the other allow for barcoding. Now the challenge, which again, I think that Philip gave a really good example of, is that clonal data is very rich in structure. So here's an example. This is from a previous paper from Dan Wagner, who was in my lab at the time, where we're using a continuous integrating barcode in a developing zebrafish embryo. And this is just looking at one clone, and it's got two subclones. And you can see that one of these subclones has a very simple structure, and the other one is distributed. So clearly there's a lot going on here. It's not that you can just look at a set of cells and say that they all have a single common progenitor, beyond the trivial fact that the whole embryo develops from a single egg. So at some point, every single cell, of course, has the same clonal origin. Now, Sam, who's going to be talking later, published a pioneering work in 2018 where she was looking at the reprogramming of fibroblasts into endodermal progenitors. And over here, I'm just giving another example of how clones can be variable. So this is a single clone which is sampled at multiple time points. And on this landscape, you can see that it's restricted to one part of this single cell embedding. But here's another clone which is everywhere. Right? So again, we have this high diversity, and these are just two of many, many clones that one could look at. So if I now just sort of take a few of these examples, and I've just added in Philip's talk right here at the end, just for fun. Here's just a few examples: different biological systems, bone marrow, fibroblast reprogramming, iPSC differentiation, embryos, heart regeneration. And each one of these has taken a slightly different strategy to the way that they think about how they're going to analyze clonal data, right? So in some cases, you just take the clones and you just look at the majority fate. In other cases, you just take the clones which are fate restricted and only pay attention to them. In another one, you throw away all the small clones, you only keep the big clones. Then there's different approaches like the ones that Philip used and ones that we've used in the past, where you look for co-occurrence or correlations between sort of enrichment of two tissues that tend to have the same clonal patterns. So what I'm going to do today is it's a pure computational talk, which is to think about maybe the way that we should approach analyzing the clonal data. And I probably won't go through all of this because I won't have the time, but the kind of questions we might want to ask, and these are the kind of questions that I like to ask, is what assumptions do we need to make when we analyze lineage tracing data? What errors are there in the experiments that we need to worry about? And then by understanding these, can we try to make experiments more effective, more interpretable? Can we work with noisier, sparse data, and therefore make these types of tools more accessible? And the work I'm going to present is really work that was driven by a postdoc in my lab. He's a Damon Runyon Quantitative Biology Fellow, Shi-Wen Wang, and it's also been funded by the CZI. So what we're going to do to try to address this problem is to root it in some mathematical thinking that Jeff Schiebinger and also some contributions from Maureen Itzan have really developed in thinking about dynamic processes, which is this notion of thinking about dynamics of states through a transition map. And the transition map is simply the probability that a cell in a particular state I will transition to a state cell in state J at a later time. And we have to be very careful about what we mean by a transition over here. We specifically mean the probability of observing a cell in state J later. And this is, I'm going to in a minute explain why this is very imperfect, but this is our starting point, and that's what I'm going to try to solve today, okay? So, oh yeah, well, I'm going to get to that point right now then. So the reason that this is imperfect is that the probability of seeing a cell in state J depends not only on the probability of I moving towards J, but also how much it divides. And fundamentally, a classical biologist would think about the difference between instructive and selective effects. Transition maps are very poor at resolving those things. So we're going to accept the fact that we are going to be able to tell whether state I is more likely to give rise to a cell in state J, but not why it's more likely. And that's problematic, but we're going to leave it there today. The good news is that if we do have these transition maps, we can start to make statements about the probability of a cell giving rise to a particular point, looking at the structure of boundaries and what are the relevant genes that change across a boundary. We can chart pseudotime, we can reconstruct hierarchies, we can look for fate predictive markers, and maybe look for putative early regulators, and say something about the potency of each state. So even though these are very imperfect, they're a really good starting point just to make some progress, okay? So what technical challenge would we have to address in order to derive these maps? So one problem is known as barcode homoplasy, which is that we can have different clones which acquire the same barcode. We can also have situations where we lose barcodes, where we lose cells from a clone. There's many tissues where we cannot resample the clone multiple times, so we only get to look at the clone once. And then there's the issue that the larger the clone is, the more it tends to be dispersed across multiple tissues. It's very practical. So just to zoom in here, what do we mean by barcode homoplasy? If we have, say, these evolvable barcodes, we gradually acquire a first barcode, a second barcode, a different barcode in another cell, but then accidentally one of these cells is acquiring the same barcode too again. And now, even though the true lineage is, as we see on one side here, the observed lineage looks like there's some coupling, and that's, of course, going to introduce an error. And these things can be much more severe. You can have a complete collapse of clones onto the same barcode. So I'm going to tell you about a method that Xu Wen has developed, and this is the sort of TLDR, black box. I don't care what it does. So there's this method. It's coherent sparse optimization. It's right sitting here in the middle. It will accept both resampling experiments, but also situations where we have early data on the transcriptomes, but only later data on the clones and the transcriptomes. And then it can also deal just with straight up sort of clone-free inference, although that's very similar to the kind of approaches that are being used today. And the hope is that we can then infer transition maps, and from the transition maps learn these other features. So the question is, how do we do this? And I should just advertise, we've just put this up online, and if you have any feedback, we'd be very grateful. It's fairly well documented. So what's the general principle? It's actually very similar to what Philip just told us, which is that if you want to learn transitions, the right thing to do is to focus on what's happening within clones. But now, rather than doing it within a single clone, you can think about looking at all of the clones in your data and constraining your transitions to maximize the transition of a clone of a cell that's initially in a particular state in T1, transitioning to another state in T2. And if you have a transition map which maximizes these transitions, it's constrained by what the clonal data is telling you. Okay, so that's essentially the idea. Now, the problem is that there's far fewer clones than there are transitions. So this is a massively under-constrained problem, and we need to somehow add more constraints. So we're thinking about reasonable assumptions here. And the first one, which is very reasonable, is to think about the fact that most cells cannot access most states. A cell in a particular state is only likely to access a few states that you observe in the system, and that is known as sparsity. So the transition map is likely to be sparse, and that can be enforced by enforcing an L1 norm on the transition map. And then the second requirement is that these transition maps should be coherent, which means that cells in very similar states are likely going to have very similar fate probabilities. So their transitions are going to be similar. So this top case is likely, and this bottom case is less likely. That's sort of the idea. And this can be enforced by minimizing the second derivative of the transition map on the spatial graph of cell states that we see. So you can put all of this together, and you get an optimization problem. And this optimization problem, we call it coherent sparse optimization. Sparse optimization is a very well-known problem. It's sort of compressed sensing, and it's being introduced into single-cell data. And the idea of enforcing coherence here just adds an extra aspect, which requires some algorithmic developments, and I won't go into that today. So the question is, how well does this perform? Let me just, so I guess that I'm going to try in the next few minutes to just give a few data sets where we've explored the performance, and then show that we can also potentially make predictions that haven't been documented before, although there's not going to be anything terribly surprising today. So there's not a huge amount of new biology here. Hopefully there's just a tool which will be useful as people start to think about these tools and might be intimidated by how to interpret the data. So the simplest thing that we could do is to just look at simulations, right? We can set up simulations and look for and recover what's happening. That's a pretty low bar, but we better get that right. We can then go back to some published data sets. So we're going to go back to a published data set from my lab, which is looking at hematopoietic differentiation from a multipotent progenitor into multiple cell types. And we resample these clones over time, and we can try to use this as a way of assessing the performance of our method. I'm going to go back to Sam Morris's data and look and see whether we can use this as another benchmark. I'm going to look at another data set from Darrell Cotton's lab, which was published last year, looking at iPSC differentiation into endoderm, and again, try to see what we can say about these data sets. Okay, and that's it. So that's what I'm going to try to give you a few highlights from each one of these in the time that we have. So the first thing is, the good news, no big surprise here, is that if we make simulations, it turns out that we can reconstruct the transition maps from which the simulations were derived very accurately. So here's an example. This is just the density of the transition map values. You can see it's sparse. Most of the time it's 0, but occasionally, you get a non-zero element. And when you try to reconstruct those elements, you can see that you do a very good job at getting them just at the right magnitude. The good news, and this is actually more interesting, is as we start to mess around with the quality of the data by gradually reducing the number of barcodes and taking clones and mushing them together, we can see that if we were to just naively take every early cell and relate it to every late cell, as we combine clones, we're going to get lots of errors because those cells were never a part of the same clone. They were never sort of part of the same lineage. And the performance decreases very, very rapidly as you increase the homoplasy. But because we're enforcing coherence and sparsity on our map, it turns out that those unwanted transitions are quenched, and we get extremely reliable and reproducible recovery of the transition map, even with pretty bad data. We can have very, very few barcodes, and we can have lots of errors, just giant clones or quasi-clones, and we still get very good recovery. So that's encouraging, and that's not entirely trivial. So now let's go back and then look at some real data, right? Because simulations are easy. And we're going to go back again to this hematopoietic data set. So there's this original data set has 130,000 cells. It's got about 50,000 cells which are clonally barcoded. And if we, again, just focus on the two single most well-covered lineages, this is neutrophils versus monocytes, this is the original data looking at every cell and mapping what it's going to do later. So the red cells are ones which will end up giving rise to a neutrophil, and the blue cells will end up giving rise to a monocyte at some later time point. We can also zoom in here. So I'm going to use this as a ground truth. Of course, it's not a perfect ground truth because it in itself has some experimental error. But what I can now do is try to destroy this data set in different ways and see whether I can still recover the data. And just to go back, this was a pretty big experiment, 130,000 cells, 50,000 of them clonally barcoded. Wouldn't it be great if we could do this with a fraction of the data? So here's what we can do. If we take the 15% largest, messiest clones, not ones that are lineage restricted, but ones that are all over the place, and we throw away 85% of the clonal data, this is what we get if we just naively take every cell and ask what it gives rise to. It's pretty messy. But now when we apply COSPAR, we can see that we can still recover this nice, smooth partitioning. So it seems to be able to preserve some of the information, even once we lose that information. The other thing that I should mention is that we can also go now back to the full data set, but look for regions where we had very little information. Like there were very few cells that in this data set differentiated into megakaryocytes and erythroid progenitors. And if we were to just apply the original methods that we developed for the paper, if we compare two alternative fates, we essentially get no hits for genes that are enriched in these clones. But once we apply COSPAR, we can get large numbers of genes which are associated with these clones. So it seems to be useful in that sense. And then the final thing I should say is that we can also just take a single time point, the later time point, and then back infer the fate potential by essentially jointly solving not only for the transition map, but also for the maximum likelihood on the distribution of clonal states at early time points. So we can see that if we only take a single time point here, we can now still get a reasonable description of the transition map. Okay, so I'm going to sort of give you the same types of highlights for another data set. So here we now are using SAM's data set as a ground truth. And now what we're going to do is start to throw away data, and we'll do this quickly. So in the first case, we start to throw away all of the high quality clones. Everything that was lineage restricted, we gradually throw away until we're down to just 10% or even fewer of the clones. And these clones are all over the place. And we can see that when we look for the marker genes that Sam in her paper predicted are associated with reprogramming, we see that we are essentially very stable in recovering those. On the other hand, when we apply the original methods, things start to go south pretty quickly. And it's not surprising because we're looking at very messy clones. The other thing that we can do is only take the last time point and again look back at day 15 and day 21 and predict what the fate biases would be by looking at the structure later and back inferring it to the earlier time points. Okay, and then finally we can even go back to the first sort of very first data that they have that Sam has in her paper, and she's going to probably blast me for this later. I wish I was after her, not before her, where we can now look at day 3 and we can find that COSPAR is predicting very strong heterogeneity in the earliest time point, which we predicted associated with fate bias. And when we actually look at the genes, they're not surprising. We can see that the transgene is on at a very high level in a subset of these cells. And then we see fibroblast genes that are on in the other set of cells. So it's maybe not so surprising, but it's sort of nice that we can pull that out. And we were pretty naive, so this for us was nice. Okay, right. So that one's for you, Sam. Okay, so I think I'm going to stop there. I actually had mentioned that we had another benchmarking on lung endoderm. I think that in the interest of time, I decided to remove that. But we're going to be putting this on bioRxiv soon, so hopefully you can have a look at it. So just to summarize what I've told you is that single-cell lineage tracing together do offer a tool for learning dynamics and identifying early fate regulators. I actually think that Philip did a very good job at motivating that better than I did. I did point out that clones can be very heterogeneous and that coherent sparse optimization, it allows us to formalize the derivation of a dynamic map from noisy clonal data. And that we've gone to some effort to benchmarking this with several data sets. And that we can even go back within these data sets and identify features that were not predicted or observed in the very first reports. Okay, so that's a computational tool, and I hope it's useful. Thank you very much.
All right, we have a question from Noah Greenwald, who says, can you give some intuition about the bad large clones that CoSPAR is robust to? Are they the result of barcode homoplazy or simply a progenitor cell that proliferated a lot and hence has a lot of progeny?
Yeah, I think I can. So there's really, so first of all, both of those things can happen depending on the experimental designs. There are certain experiments, and Philip's Linnaeus is a good example of that, where scarring tends to lead to stereotypical barcodes appearing. And that can often be a problem. And if I recall correctly, much of the effort of algorithm development for analyzing those data sets was to try to identify frequent barcodes and eliminate them. So that's a very practical and real problem that can occur in particular designs. The other challenge is that there's a trade-off between looking at a clone very early in order to nail the early state of a cell and letting it grow enough so that you can be sure that it has progeny, which you can then resample later. So another reason that you can have very dispersed clones is simply because the barcode integrates very early. And in the embryo, it's very easy to appreciate why that would be the case, right? If you're barcoding by the 1,000 cell stage, at that point, a mid-blastular transition is just kicking in. Zygotic transcription hasn't even started. These cells are essentially very similar, and they're just being separated spatially. So you're going to have a situation where there's barcodes that are giving rise to many, many tissues. And that's probably going to be stereotypical of many situations.
Great. I might ask my own question. This is it's a little bit of a visualization question and a little bit, I guess, of a CoSPAR method question. You have this coherence constraint. Like in practice, like how tight is that? Like how strongly do you require that these are going to, follow the trajectory, that you have a particular 2D trajectory? Because as you nicely said at the beginning of your talk, you change the parameters a little bit or different subset of cells, you can get wildly different trajectories in some cases.
So what we've done is we've stress tested this by starting off, let's say, take the hematopoietic data, and you can the cells are still the same cells, but you can take different ways of measuring distances between them. And that's, of course, going to change the way that you do this, the coherent mapping. And you find that if you then just apply a clone-free analysis, you can get quite a bit of variation in the quality of the results in the fate prediction that you get when you compare. Once you add in the clonal data, it tends to iron that out. It's really so, but you do need to have some structure there, right? So if this is integrating lineage tracing with single-cell RNA sequencing, if there's no information that's useful in the smoothness of the structure, then you're really lost. But what this does is that it takes you and it nudges you in the right direction. And this is maybe very similar in spirit to what Philip was saying, but this just formalizes the idea, right? That yeah. So.
Right. Long Kai says, great talk and great hair. Is it better to do experiments with continuous lineage tracking or do cloning tracing from multiple initial time points?
That's a very good question. Thanks, Long, and thanks for the hair comment. I would say that there's it really depends on the experimental design. In an ideal situation, if you had really perfectly evolvable barcodes so that every cell cycle you could be sure to add in additional lineage information, I suspect that it would be superfluous. You would still want to sample your clones at different time points, but it may be superfluous to start labeling at different time points because you're essentially doing that. So I think that the devil is probably in the details there. If you're limited and you just have to let the same barcode propagate through the system, then you might still be able to learn a lot of information as we've shown. But there's also a strong argument for starting the labeling at different time points so you can be sure that your cells are in different conditions. You don't have these highly dispersed clones.
Great. Maybe we have time for one more question. And so this is actually, Arjun had a question in the moderator chat, and this I'm going to combine it with this other question, which says, essentially it's like, what are the failure modes? At what point does COSPAR fail in prediction? This is from Geethika Arakatla. For example, maybe HSC differentiation is well defined, but in less defined systems, would a lack of sort of clear branch points cause issues? And then actually merging that with the third question, like what about complicated situations like trifurcations? Like, can you handle those?
Right. So I love that. And first of all, like that's totally out of like showing how methods fail is like critical. And I totally did not do that. So thank you for calling me out on that. I appreciate it. Let's see. So trifurcations shouldn't be a problem because there's really no constraint in these transition maps about the number of states. And in fact, you can also have loops in them. There's really, it's sort of very general. There's no imposition of hierarchy here. The question about failure modes. So one thing that we've found very challenging is when you have huge batch effects between time points, that can be challenging. The other thing that goes without saying is that when you're back propagate, so I went through this very quickly, but there's two experimental designs, one in which you get to see the clone twice, which requires some privilege to do. I know that, let's say, Arjun's experimental design, Sam's experimental designs, and the ones that we did in hematopoiesis have allowed it. In many tissues, like what Philip was showing, you can't dissociate the embryo and then put it back together and see the rest of the dynamics. And in those situations, you're essentially using the clonal structure that you see later to predict heterogeneity that is relevant earlier. And there are certainly ways that could fail. And in those situations, one has to take this as a hypothesis. Whereas when you have 2 time points, it clones at 2 time points, it's a much, much stronger constraint. And there's much less room for failure in a situation like that.
Great. Thanks so much for Alon. Great talk.
Thank you very much.